Hi, my name is Kim Cardoza, and I'm a protein synthesis expert from Johns Hopkins Medical Institute. Now, today I will be explaining to you how protein synthesis works. Well, first you need to understand why protein synthesis is important. Well, in each living cell, there is proteins, and proteins are necessary for cells to function. Now, the way that protein synthesis works in the human body is that it goes through two processes, transcription and translation. Now, if you have a hard time remembering that, all you need to do is remember that the C in transcription comes before the L in translation. Today I will be showing you a video and narrating the parts of how this happens, and then I will also go into more detail of how the prion could be made during this process, since a prion is a malfunctioning protein. Transcription is basically what it is, the DNA being transcribed into mRNA in the nucleus. We are looking at the gene part of the DNA, consisting of all the necessary components to carry out protein synthesis. Here we can see the DNA strand currently being split by the RNA polymerase, which is an enzyme that breaks down the hydrogen bonds between the double helix bond, or simply unzips the DNA, and then moves on to use another RNA polymerase to take a copy of the DNA strand to make messenger RNA, or mRNA for short, which will be then used to make protein later on in the process of translation. Once this is finished, the mRNA is then transported by transport RNA, or tRNA, out of the nuclear pores to start the process of translation. The mRNA is transported by the tRNA to find a ribosome, which is made by a small enlarged subunit to start producing protein. Each set of the three bases in the mRNA is called a codon, and each codon calls for a specific amino acid to be brought into the ribosome. The way that the amino acids reach the ribosome is through tRNA, as it comes attached with an anticodon, which matches the base pair as its complementary base pair. Once the anticodon meets with the codon, the nucleic acid is released back into the cytoplasm of the cell, and the process repeats until enough amino acid is made to form a protein. When enough amino acids are made, the line of amino acids will transport into the Golgi body, which will pack into the correct form to produce a protein that will be then shipped out to the designated place. So now you know the process of protein synthesis. Now we're going to move on to talk about how prions may possibly be made through this process. Now, as you may know, or as you might not know, prions are actually malfunctioning proteins. The way that our scientists at Johns Hopkins Medical Institute think that are being made in nerve cells in the brain is through the malfunctioning of the Golgi body, since the Golgi body packs up these amino acids into proteins. Or maybe what is happening is that the proteins are being shipped out to different parts of the nerve cell that it isn't supposed to, and that's the reason why it's malfunctioning. Another theory that our scientists have at Johns Hopkins Institute is that the anticodons are being paired with the wrong codon, so there could be a malfunctioning between the two. Now, the idea of the prions already being there, or maybe the DNA being malfunctioning, can be ruled out, because that would have to be something that would have been inherited. And the zombies that are in this apocalypse have just spontaneously arise. So these are the three theories that we came up with. Hopefully you can agree with them. If not, we are still working to find more theories to solve this issue.